Explain the major mechanisms underlying propulsion and mixing in the GIT tract. This topic's quite broad, and when you get into it, it gets really complex. So I'm really going to try and present the core knowledge that I think we need to know, or that I need to know, or whatever, without dumbing it down too much. So here I go. Now, the first thing you need to know is about certain properties of the gut. Now, it's got two special properties that are important to know. Firstly, it's that there are two layers of muscle. There is longitudinal and circular. Now, if we draw a piece of small intestine, for example, here, now the longitudinal muscles are arranged lengthwise, like that, so that when they constrict, they shorten the length of GI tract, whereas the circular ones are more, if you can imagine this as a 3D structure, as a pipe or whatever, they're more, they have the effect of shortening the diameter or closing or constricting the lumen, so decrease lumen size. Circumference. Yep. So that's the first important thing to get, which means that when circular contracts, it can actually do cool things like make the muscle go like that, for example. So that's that. Now the other important thing is to understand the concept that there's something called slow wave membrane potentials. Now I'm just going to draw. Now what these are is they're spontaneous waves of membrane potentials that are built into the muscle fibers. So to just draw this, okay, so the first thing you have to keep in mind is that there is a threshold for muscle action potentials. Yeah. Okay. Now the second thing is that there are these waves that don't actually reach this action potential. So these are the slow wave membrane potentials, and they're built into the muscle fibers, and this is all thanks to, or it's all done by, these special cells called interstitial cells of Kajal. So they cause this membrane potential to occur in a slow wave polarizing depolarizing form. So that's important. Now the next thing is that when there is a stimulus or an input to the muscle, such as a large amount of food in the small intestine, that's gonna, so no input, I should say. Now when that input is gone up, like this, we'll say, so you have input in, you want the muscles to be doing something. The input is constant, see, constant input. But you don't want there to be a constant constriction of the whole small intestine. You want it to happen in pulsations, so it can knead and push and mix your food or chyme around. So what this means is that this is kind of superimposed on top of the slow waves. So you can imagine you add some distance maybe not, I didn't add it quite enough, but so you add it. So that, we'll just draw it over the top here. So you can see that with a constant stimulus of contraction, it doesn't, a constant input does not equal constant contraction of the muscles. So now if we draw in yellow muscle tension, so we draw in yellow here. Oops, I can't draw very well. So if we draw an arrow here, we'll see muscle tension occurs in pulsations or waves. So what this means is that with a constant stimulus, there's still waves or pulses of muscle contraction. And this concept is really important and it's key. And this all happens largely thanks, but not only, but mostly because of the interstitial cells of Kajal. Okay. So now we've gone through the basics, which has taken 7 minutes. I'm now gonna talk about what happens when you eat. So we draw a hamburger here. Yeah, there's our hamburger. Here's our mouth around our person. So here's our hamburger. Here's me, esophagus, going down to the stomach, duodenum. Alright. So what happens when we eat to the stomach first? First we'll talk about the stomach. The stomach has two main purposes, to store food and to empty it into the rest of the gut. So first of all, storage. How does that happen? Yeah, you'd think, oh yeah, pretty simple, just goes in there, whatever. It's a big pouch. There's a little bit more to it than that. First of all, what happens that when you have input from your esophagus, so input from esophagus will go to the hindbrain. This is all vagal. And then down vagus will send signals to inhibitory motor neurons. So I'll talk about excitatory motor neurons a little bit later, but right now we're talking about inhibitory motor neurons. So it sends it to them, and what this does is, surprise, causes relaxation. It inhibits muscle contraction, and this is called receptor relaxation. Yep, cool. Now the other important thing that happens is that so it stimulated the esophagus. So in the stomach, there's chyme in there. That causes outward pressure, distension. So the next part is that distension itself activates an accommodation reflex. So this is a local thing. Accommodation reflex. And this will also inhibit muscle contraction. Yep. So that's storage. Now let's talk about emptying. Gastric emptying. How does this work? So this is really important. Gastric emptying. It's really important. Why is it important? Because what comes out of the stomach? What is your chyme? Some of the properties of it is that it's high in acid. It's very high in osmolarity, and it's high in nutritional content. So what happens if you have a big meal and the stomach just empties everything out in one big hit? You're gonna have heaps of acid put into the duodenum, and that's going to cause things like ulcers, which will then lead to bleeding out, which will then lead to death. Osmolarity, you're going to have all this salty substance in a very absorptive area, which means that all your water from your blood and all that is just going to enter your GI tract, and you're going to have huge diarrhea and water loss, which will then lead to death. And decreased nutrition, if every time you eat, it just all of it goes into your GI at once, you're not going to be able to absorb it all. So you're going to not be able to absorb, decrease absorption. It might all get stuck as well. Also blockage, which can also lead to death as well. So gastric emptying is really important that it's done properly. So how does it work? So again, I'll draw the stomach. What I'm drawing here is the pyloric sphincter, right there. Pyloric sphincter. Now this is really important. So what happens is that in the fed state, you're going to have slow waves. Now these are normally around 3 per minute. Now they contract, so you remember the slow waves I talked about before. Now they contract, originating in the upper corpus, so that's the body of the stomach. So originating here along a greater curvature here, and descending down into the antrum here. And what this does is it will send your food down into the pylorus, and through the pyloric sphincter into the duodenum. This is the duodenum here. Alright, so that's what happens. So slow waves causes phasic pumping. Remember, it happens in phases. Pumping of contents through the pyloric sphincter, which I'm going to even do in a different color, which is normally weakly closed. Now that's important. It's normally weakly closed, which means two things. Firstly, not all of it is partly closed, so not all is going to go in. So some is actually going to not actually fit through the sphincter and it's going to get sent back. And this is called retropulsion, which I'll talk a little bit more about in a minute. Now, the other thing that happens is that it will little amount enter duodenum. Now, what happens here? A little amount of what? So let's remember that it's a little amount of your acid, a lot, quite acidic acid, high osmolarity, and high nutrition. Alright, so these three things are sensed by the duodenum, and what happens then, sensed, is that it begins a negative feedback which causes the strong, remember up here I said weakly, the strong shutting of the pleuris. Now that's going to cause more retropulsion. Now, as I said, retropulsion, by having the contents go back up into the body of the stomach and sending them through, is going to mix further, and it's also going to break up any large particles, or at least help do that. So that's important. Now, the strong shutting of the pleuris will also, this is getting a little bit, go this way. Now, will also decrease, I'm going to change colors now, will also decrease the amount entering the duodenum. So it's going to decrease that. Now, so because that's decreased there, that's also going to inhibit, or the negative feedback's no longer going to be happening. Does that make sense? Yep. So less negative feedback, that's not happening anymore, which means that the pleuris is going to go back from being strongly shut to weakly closed, and then the cycle happens again. It's a little bit messy, and I apologize, but that's what happens. Alright. Now, there's another little thing that's, I think, worth mentioning, is that the nutritional content especially, I've drawn this yellow. Why have I drawn it yellow? To signify nerves. So the nutrition is also, the high nutrition content is also sensed, and this causes another thing. Vagal afferents will then go to the hindbrain. Yeah, so especially if you're eating really dense food, and this is gonna cause vagal, and then send out its vagal efferents. Now, this has the effect of slowing down these slow waves. So in the stomach, so it has that effect on the stomach, and then that's also gonna decrease the slow waves, decrease the phasic pumping, decrease the content going through, and just slow that all down. And then it also has other effects on the whole GI, other effects on the GI tract. So you can think vagus is parasympathetic, rest and digest. So as a whole, in other areas, it will actually speed things up a bit. But overall, this is gonna this has the effect to cause a consistent calorie load, which I'll just write somewhere wherever I have room. Consistent calorie load. There we go. So that's the stomach finished. Through these mechanisms, basically it will send a consistent calorie load to the small intestine. So now let's talk about the small intestine. Now, the small intestine has three key functions when it comes to the movement or motility. It needs to mix the contents with the secretions and all that kind of stuff. It needs to expose the contents to the absorptive surfaces, and it also needs to propel the waste and leftovers into the larger intestine, where it can then be excreted. So that's the three key things that movement of the small intestine have to do. Now, the mixed and expose is mostly done by something called segregation. Now, if you think segregation, think sausage or sausage links. Yeah. what happens here, and there's a few good animations if you Google, is that in segmentations you will have, so here's a relaxed bit of small intestine, and then here's a segmented part of small intestine. Now, so you can imagine here is circular muscle contraction. Now, so what happens in segregation is you have alternating points of circular muscle contraction and distension. So that construction will cause distension proximal, both in front and behind. Now, this distension proximal will then this distension will then have the effect of, in the distended area, the muscles here, the circular muscles here will then contract. So that circular muscle there will cause distension there, which in turn causes more muscle contraction. And this happens in waves, just like I talked about at the start. If you remember, just zoom up quickly. So all this stuff here is quite key. So then that will cause muscle contraction. So this will look like, this is a bit messy, I apologize. I'm not the best drawer. Especially when it's in real time. So you can see it alternates. So alternating. Now what this does is it needs the chyme, or whatever you want to call it, food, but it doesn't propel. No propel. Or it might propel a little bit, but it's quite minimal. So that's kind of the mixing and the exposing. It happens in segments. So you just think of the length of sausages, and then imagine squeezing the sausage and then releasing, and it shouldn't be too hard to visualize, but my drawing's a bit piss weak. Anyways, the next thing is something that's a bit more fun to explain, and that's the peristalsis. It has a lot of attention in the books, and they talk about the textbooks will often talk about how it's involved in the mixing and that a lot as well, which isn't really that true. Peristalsis is all about propulsion when you need it. So let's draw. It's not a good color. Let's draw out a segment again. So imagine this as a segment of your small intestine. Now, with here being the lumen and there being the muscle wall. Okay, now peristalsis works like this. Now, there are two types of muscle fibers. Two types of motor neurons. There are inhibitory motor neurons, and there are, I'll draw these in green. There are excitatory motor neurons. Yeah, that's two important concepts. Now, what else is important is that the excitatory motor neurons project orally. So excitatory motor neurons mean that they have an excitatory effect on muscles, and they project orally. So if we imagine the mouse here, oral, and the anus is this way, anus, yep, they project orally, and the inhibitory motor neurons project anally. So let's draw this out. So if we imagine here's a inhibitory motor neuron, and it projects this way, and here's another one projecting that way, and another one projecting this way. Now if we draw excitatory ones, we'll draw one here, projects this way. Yep, orally, making this point really clear, I hope. So when these are all stimulated, so imagine here is a stimulus, a bit of chime that's gotten backed up or condensed through, say when the segregation goes a bit wrong, or it might be gas, or it could be what's quite important and one of the probably evolutionary, very important purposes of peristalsis is, say, something like bad bacteria that shouldn't be there, or toxins that we need to get out of our body. So what this does is it stimulates all of them, stimulates all the neurons, with the excitatory motor neurons being oral. Now that's going to cause a contraction on this end, oral, and the inhibitory ones are also going to be stimulated, which is going to cause relaxation of the circular muscle fibers this way. And then that's going to have the effect of sending whatever stimulated these fibers, whether it was distension or bacteria, to travel anally. And this peristalsis will propel fluid or bacteria or chyme or more solid substances quite quickly towards the anus. And that's really important. So through peristalsis and segregation, you will have food going from the small intestine to the large intestine. And the happenings of the large intestine aren't really affected by being in the fed or fasted state so much. It's kind of a different, longer process. Whereas your feces will stay in the large intestine for days, whereas the small intestine is more hours. So I've talked about what happens when you eat. So now that we're at the point where the food is being taken to the large intestine, what happens when you are in the fasted state? Okay. Now in fasting, there's one more important motility concept to understand, and that is called the migrating myoelectric complex. Alright, migrating MMC. Alright, 3 stages, 1, 2, 3. One's little activity, cool. Second is irregular activity, and the third is maximum activity of all slow waves. So if we go back, in this point you can think of all these slow waves are maximally contracting or maximum activity. Alright, so what's the purpose of this? Well, actually, first of all, I'll just say it happens every 80 to 120 minutes. And the purpose of it is really, it's kind of the housekeeper. I'm just going to write clean up, VGI. especially the small intestine plus stomach. So you don't want bits of bacteria sitting around in a unmoving environment growing, and you also want to get rid of any dead or sloughed off cells and all that kind and secretions, especially considering some of the secretions will damage your own cells if there's too much of them. So that's important, and that's why you have it, and that causes kind of a flushing out of sometimes your stomach and especially your small intestine. Now, how exactly does it works is not very well known, and what is known about it is kind of out of the scope of this video. But I will mention that it is interrupted by three key things: food, or and anesthesia, and also a phase 3 of this. So part 3 of it will interrupt another part 3. So it can only have one phase going at one time. So that's the three important things. So that's about it. I'll see how long have we gone for. This has been my longest video. I thought it would be simple, but there you have it. Long video. That's it. I'll talk about the large intestine in another video. So there we go.